Over the next 2 days, we'll go through the process of transcription. Probably we'll do that on Thursday, take it apart completely. And then on Friday, we'll take translation apart completely. But for today, I want to just give you kind of a brief overview of what's happening in each of these.
Has that really expensive machine come yet?
Do what?
Has that expensive machine come yet?
No, it hasn't. Okay, so transcription. We saw just a while ago from the pictures that it will result in the synthesis of messenger RNA, which we abbreviated as mRNA. And if we make the assumption that this is a eukaryotic cell, then we could say that this event is going to be an event exclusive to the nucleus. If it's a bacterial cell, then it's going to happen wherever the DNA is, which is not in a nucleus, certainly if it's a prokaryotic cell. And we know that the recipe for creating that mRNA is based on DNA, so we could say for sure that DNA serves as your recipe book or as a template for that mRNA strand. And this is why to me it gets confusing between 16 and 17, the chapter we were just on, is right now. And that's because the major enzyme of this process is called RNA polymerase. In the last chapter, we had spent some time working with DNA polymerase, and that obviously is different than RNA polymerase. Remember, DNA polymerase made DNA strands, correct? So RNA polymerase could be expected to make RNA strands, which is what mRNA is. Good? Then translation, right after transcription, is going to result in the production of your protein. And we saw that this event was always exclusive at the ribosome. That's why all cells have to have ribosomes. They also all have to have DNA. Things that are living must meet those two requirements, really as a minimum. And the mRNA that was created by transcription serves as your foundation or your template for the production of that protein. So the order of amino acids, that going back to that primary structure of a protein, that is based exclusively on what was coded in your DNA, which was encoded in your mRNA strand. And we'll see probably on Friday that there are several important enzymes that are involved in this process. Now, assuming that this is a eukaryotic cell, we could agree that there was a separation of these two events because the transcription was only in the nucleus. The translation was on the ribosome, which is going to be for sure outside of the nucleus. So we could say that between these two events, assuming it's eukaryotic, which most of our cases will be, that the nucleus is going to cause a separation of these two events in time and in space. So because one event's transcription is occurring in the nucleus and the other event is occurring outside the nucleus, there's a division of when these two can happen. It'd be analogous to saying that you go to the library and you transcribe something from an encyclopedia, you create some notes, and then your notes go home and you use the computer to take those notes and create your paper. Your paper is what shows the teacher your potential. Agreed? If you turn in your note cards, does the teacher give you anything valuable? No. Like you can't, your mRNA, in other words, your transcript, your copy of the notes doesn't facilitate anything for you. It doesn't represent your change. It doesn't represent your grade. What represents your grade is the paper, right? So I want us to, it's the same principle. You find a resource, you copy the resource, then you use the resource to create your final product. Right? I find a great recipe in the recipe library. I copy it down because I can't take that book home, and I use it to create my sandwich. Does Betty Crocker morph into a sandwich each day for you? That would be cool, but it doesn't happen now, does it? So that means that you have to take your transcript and you have to cause it or allow it to create a protein for you. That has to serve as the resource or the template for your protein. Okay. Last couple little things real quick that I want to go over. Just two last little topics and then we can be done. This process that we've been discussing is known as the central dogma of biology. This large theme that describes how your DNA is used to create mRNA and several other forms of RNA for that matter. In order to create your protein. So your DNA provides the resources, the information for the production of your final product. And that's through the reactions of transcription, or the events, I should say, of transcription, and through translation. Good? Okay. Let's remind ourselves a little bit about the monomers of both of these guys. What's the monomer of DNA? Okay, so the monomer is the nucleotide. What's the monomer of a protein? Okay. How many different nucleotides are there? 4. How many different types of amino acids are there? Just 20. 20 different types. Can I name them all? Maybe. We'll have this contest after, in just a second. Myself. Okay. Now imagine, and I want you to think about this real quick. Would it be possible for one nucleotide to give rise to a single protein? That wouldn't work out because you only have 4 nucleotides, so that could only account for 4 of your amino acids. I'm sorry. Right? What if you use 2 nucleotides? That would make 2 choices, 4 apiece, so that's what, 16 different opportunities or 16 different outcomes. That can't happen. So it would take 3 nucleotides, an ATGC here, an ATGC here, and an ATGC here. How many different combinations can you make? 64 different combinations. So we want to get this concept that it would take 3 nucleotide bases to code for one single amino acid. These 3 bases that are used back to back to back, these 3 that occur and represent one single amino acid, is called a triplet codon. And you might have seen the word codon before. If you look at a chart like this, this is a codon chart. These are the codons for mRNA, or just simply RNA. If you have AUG, that corresponds to a specific amino acid. The first letter I mentioned was A. So you know once this says the first mRNA base, so we would know we got to be in here. The second letter was U, correct? So you look at U, so now we've got to be right through here. And then I said it was AUG, right? So we've got to be this one. That goes for methionine, which is always your start codon. That means that whenever that sequence, whenever AUG is on mRNA, that tells the ribosome that you've got to start translation at this point. It's your start sequence. And you see that there's listed up here 3 different stop sequences, right? UAG, UGA, and then UAA. All those are your stop sequences. Right? So you can see here that there's different codons corresponding to different amino acids. Since there were 64 different combinations but only 20 different amino acids, do you know what this means? There's going to be some redundancy. Look at the first box. PHE up top. PHE stands for phenylalanine. It's an amino acid. There's 2 ways to code for phenylalanine, right there. Makes sense. On some amino acids, there's more than 2 ways. Sometimes there's 4. Serine right next to it has 4 different ways to make it. So we can agree that actually in serine even has more if you look down here. So this is going to allow for some redundancy in codons and the formation of amino acids.